Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing DBT JRF paper 2013. I have done it in two parts and this is the first. So coming to the first question. Mammalian cells in primary culture experience half-life limit after 50-60 cell generation time. However, in a rare situation or employing certain agents, cells can be induced to immortality. Which of the following genes or proteins have a major relationship with this? So before coming to the question, what is half-life limit? So half-life limit states that normal human cells can only replicate and divide 40 to 60 times before it cannot divide anymore and will break down by programmed cell death or apoptosis. So they have asked mammalian cells in primary culture. They experience half-life limit after 50-60 cell generation time. But if you add certain agents, they can be induced to immortality. That is, they can keep on dividing. So which of the following genes or proteins have major relationship with this induction of immortality? So it is P53. So P53 is a gene that codes for protein that regulate cell cycle and hence function as tumor suppressor gene. Once tumor suppressor gene is mutated, the cell becomes immortal. So the answer here is option C, that is P53. Then next one. Snare proteins are found in the membranes of all of the following compartments except mitochondria. Answer is option A. Then next, in the preparation of golden rice, a few exogenous genes have been used in order to achieve the production of vitamin A through recombinant DNA technology. This is an example of metabolic extension. Next one. All of the following enzymes are linked to reduction of NADH except succinate dehydrogenase. It converts succinate to fumarate and FAD is reduced to FADH2. So NADH is not involved. Then which of the following are components of a phospholipid? So they are fatty acid, phosphate group and glycerol. Answer is option B. Which one of the following is a signaling receptor? So the answer here is option B, that is toll-like receptor. That is a signaling receptor. Then next one. Amino acid analysis of 1 milligram of pure enzyme yielded 58.1 microgram of leucine. Molecular weight 131.2. What is the minimum molecular weight of the enzyme based on leucine content? So weight of leucine by molecular weight of leucine is equal to weight of enzyme by molecular weight of enzyme. So substitute and calculate for X. You will get 2258. Answer is option D. Then next one. The first metabolic intermediate that is common to the aerobic metabolism of glucose and fatty acid is. So it is the common metabolic intermediate is acetyl-CoA. Next one. While sedimenting a microparticle by centrifugation, instead of increasing the RPM to attain the desired G force, what else can be done to attain the complete sedimentation? So for that you can decrease the density of the medium. Answer is option A. Then for passive vaccination, which antibody will be most appropriate? Naturally, polyclonal antibody. Next one. Which of the following is the best method to determine the phospholipid asymmetry in a plasma membrane? So they have mentioned it is phospholipid asymmetry. So phospholipids can be determined using thin layer chromatography. Answer is option D. Prosthetic groups such as iron sulfur clusters and heme function too. They both accept and donate electrons during electron transport. Answer is option C. APS or ammonium persulfate is used in SDS page for free radical formation. APS is for free radical formation. Answer is option D. Then next one. Which of the following statements about tryptophan operon is not correct? It is normally turned off if tryptophan is present. Yes. Tryptophan acts as a co-repressor. Yes. The regulatory gene product is inactive by itself. Yes. Tryptophan binds to the repressor protein and inactivates it. Tryptophan binds to tryptophan repressor and causes it to change shape, converting it into active form, that is DNA binding form. The tryptophan repressor with the bound tryptophan attaches to the operator, blocking RNA polymerase from binding to the promoter and preventing preventing transcription of the operon. So the answer here is option D, that is incorrect. Then glucose labeled with C14 at C6 is added to a solution containing the enzymes and cofactors of the oxidative phase of the pentose phosphate pathway. The radioactive label will be observed at. So this is the glucose 6-phosphate is oxidized to form ribulose 5-phosphate. And a carbon is removed as carbon dioxide is released. So this is glucose. It is phosphorylated at C6. It is phosphorylated at C6, that is over here. Not phosphorylated, 1 minute, is labeled with C14 at C6. This carbon atom is labeled. And after NADP is getting converted to NADPH and carbon dioxide is released, you get 5 carbon compound, that is ribulose 5-phosphate. And where will the C14 be labeled? It will be labeled at carbon number 5. This carbon is removed as carbon dioxide. So it will be at carbon number 5. The labeling will be at carbon number 5. So carbon number 5 of ribulose 5-phosphate. Answer is option A. The molarity of 15% NaCl is, so 15 gram of NaOH in 100 ml of the solution. Molarity is number of moles of the solute by volume of the solution in liter. So moles of NaCl, mass by molecular mass, 15 by 58.5. Molarity is equal to 15 by 58.5 into 0.1. 0.1 is the volume. 100 ml, so convert it into liters, you will get 0.1 liter, and molarity, if you substitute in the formula, you will get 2.564. So answer is option A. Then next one. Which of the following antibiotics is used to demonstrate the fresh protein synthesis in response to challenge by an inducer? It is chloramphenicol. Tetracycline is not used because it inhibits initiation itself. So chloramphenicol is used. Then identify the mismatch. Alkaline phosphatase removes phosphate group present at 5' end of DNA. That's correct. DNA polymerase 1 in Nick translation. S1 endonuclease cleaves only single-stranded DNA. DNase 1 cleaves only double-stranded DNA. It cleaves both single-stranded and double-stranded DNA. So the mismatch is option D. Then sciophytes are plants that prefer to grow in shade. Answer is option B. Which one of the following is a micronutrient of plants? Manganese, Mn. Answer is option A. Hell's Gate 12 is an RNAi vector. Answer is option C. In protein A, glutamic acid is replaced by glutamine to make protein B. Which technique can resolve these two proteins? So, glutamic acid, it is replaced by glutamine. Now, isoelectric focusing can be used. It separates protein according to their isoelectric point within a continuous pH gradient. Now, glutamic acid is acidic whereas glutamine is not. So the pI also will be different for glutamic acid and glutamine, so it can be resolved by isoelectric focusing. Then genes related through the descent from a common ancestral gene are called as paralogs. The Bohr effect in hemoglobin refers to reduced affinity for oxygen at lower pH. Answer is option D. In a biological system, amplification of a signal is a fundamental regulatory principle. Which one of the following is not an amplification system? Blood clotting. Yes, it is amplification. Complement activation, that is also an amplification system. Transmembrane receptor mediated gene expression in biosignaling, that is also an amplification system. Ciliary movement in cochlea, that is not an amplification system. Answer is option D. Then next one. The most pleiotropic colony stimulating factor is, so it is interleukin 3. Answer is option C. Then which one of the following cells will migrate after injury in the central nervous system? So it is microglia. Answer is option A. Then next one. Pseudo-unipolar neurons are found in, so they are found in dorsal root ganglion. Answer is option B. The genetic map of 3 genes A, B, C is as follows. AB is 10 map units, BC is 5 map units, AC is 15 map units. If an individual with genotype ABC, the percentage of gametes expected to be capital A, capital B, capital C is. So this is the genotype that they have given, ABC, and here is ABC. Now there will be a crossing over between capital A and capital B as well as capital B and capital C to produce the gamete capital A, B, C. So probability of crossover between genes capital A and capital B is 10%. 10 map units is equal to 10% recombination frequency. So 10%. Capital probability of crossover between genes B and C is 5%. That will be 1 by 20. Now percentage of gametes expected to be capital A, B, C is probability of crossover between genes A and B into probability of crossover between genes B and C. So 1 by 10 into 1 by 20, 1 by 200. If you convert it into percentage, you will get as 0.5%. So the answer here is option A. Then Mendel's law of segregation as applied to the behavior of chromosomes during cell division means that alleles of a gene separate from each other when homologs separate in meiosis 1 or when chromatids separate in meiosis 2 if there is a crossover between gene and the centromere. Answer is option C. Then next one. Which one of the following markers in the brain is used for diagnosis of rabies? That is Negri bodies. Answer is B. Which one of the following cell type is the most characteristic component of the early stage of acute inflammatory reaction? It is neutrophils. It is in the early stage they have asked for inflammatory reaction. It is neutrophils. Then competitive inhibition of an enzyme by a competitive inhibitor can be overcome by simply increasing the concentration of the substrate. Answer is option B. There is a sure shot question from competitive inhibition I have seen for majority of the entrance exam, not only for DBT but also for other university PhD entrance exams also. There is one question from competitive inhibition. Majority of the question papers will have that question. Okay, next one. In which of the following reactions is the unit of rate constant and the rate of reaction the same? So rate of reaction and rate constant, the unit is moles per liter per second. So it is same for zero order reaction, because for zero order reaction rate is equal to K concentration of A raised to 0, that is equal to K, where K is the rate constant. So rate of the reaction is equal to rate constant in the case of zero order reaction. So The unit of rate constant and rate of reaction are the same in the case of zero order reaction. Then next one. In an enzymatic reaction, if the enzyme concentration is increased from 1 mg to 2 mg, which of the following statements is correct? So, the enzyme concentration is increased. Enzyme concentration is increased means the Vmax increases when enzyme concentration increases, but Km does not depend on enzyme concentration. So Km will remain constant but Vmax will be more. Answer is option C. Then which of the following receptors perceive blue light in plants? So cryptochrome, they perceive blue light in plants. Paralytic shellfish poisoning is caused by consumption of molluscan shellfish contaminated with saxitoxins. Answer is option C. The site of production of gonad inhibiting hormone in crustaceans is X organ. Answer is option C. The immunoglobulin isotype found both in cartilaginous and bony fish is IgM. Answer is option B. In Bhopal disaster, by which hazardous substance the toxicity effect get increased? It is by methyl isocyanate. Answer is option A. Which one of the following microorganism can be effectively used as a biocontrol agent? Bacillus thuringiensis. Answer is option A. Which of the following gases contribute most to the greenhouse effect? It is carbon dioxide. Answer is B. One of the best represent the central dogma of bioinformatics. It is sequence, structure and then function. That is the central dogma of bioinformatics. Then multiple sequence alignments are not used to derive HMMs, that is hidden Markov models. HMMs are hidden Markov models. So multiple sequence alignment are not used to derive hidden Markov model. Answer is option D. Which of the following matrices have been used to identify distantly related homologues? So they have asked for distantly related homologues. So for closely related sequences, you have to choose Blossom matrices. 1 minute. For distant related sequence, they have asked for distant related sequences. So for distant related sequence, select low value Blossom matrices, that is Blossom 45, or high value PAM matrices, that is PAM 250. For distant related homologues or distantly related sequences, you have to choose low Blossom matrices. So here low is Blossom 45, so you have to choose that. If it was PAM, then you have to go for PAM 250, or I think that is the highest, PAM 250. Then next one. Among the different amino acid side chains in proteins, which of the following pairs might form side chains hydrogen bonding interaction with each other? Among the different amino acid side chains in a protein, which of the following pairs might form side chain hydrogen bonding interaction with each other? Okay. So side chain hydrogen bonding can be formed between asparagine and serine. Answer is option C. If a protein is known to bind Ca2 positive ions, which one of the following side chain is likely to be involved in Ca2 plus binding? It is aspartic acid. Answer is option A. Then next one. Which one of the following suggests the multimeric nature of P53? So the answer here is dominant negative mutants are available. Now, dominant negative mutants. So dominant negative mutation, a mutation whose gene product adversely affect the normal wild type gene product within the same cell. That is a mutation where the gene product, it will affect the normal wild type gene product within the same cell. That is a mutant will affect the wild type gene product within the same cell. That is called as dominant negative mutants. Now, dominant negative mutations are most effective for proteins that need to assemble into multimers to be functional. So, dominant negative mutation, they are effective for proteins which are multimers, and they have asked the multimeric nature of P53, so it is dominant negative mutants. And physiologically active form of P53, it consists of a tetramer of 4 identical amino acid subunits. And mutant P53, it exerts a dominant negative effect by preventing the wild type P53 from binding to the promoter of its target genes. So, which of the following suggests multimeric nature of P53? It is dominant negative mutants. Then which of the following is also called suicide enzyme? So, it is adenosine deaminase. Answer is option C. Then IgG in patients with rheumatoid arthritis has abnormal. It has abnormal glycosylation in case of rheumatoid arthritis people. Then which one of the following pathogen does not use antigenic variation as a major means of evading host defense? So influenza, HIV, trypanosomas, they all have antigenic variation, whereas top of form Streptococcus pneumoniae doesn't have antigenic variation property. So answer is option A. Warburg effect is characterized by. So Warburg effect, it is an increase in the rate of glucose uptake and production of lactate even in the presence of oxygen. Usually glucose is converted to lactate in the absence of oxygen, but here in case of Warburg effect, there is increase in the rate of glucose uptake and production of lactate even in the presence of oxygen. So answer here is characterized by increased glycolysis. Answer is option A. Then Amanita phalloides, which is responsible for the majority of fatal mushroom poisonings, contains alpha amanitin that specifically inhibits RNA polymerase. Answer is option B. Then phenyl methyl sulfonyl fluoride is A or an. It is PMSF. It is a irreversible inhibitor of serine protease. PMSF is an irreversible inhibitor of serine protease. An irreversible inhibitor inactivates an enzyme by bonding covalently to a particular group at the active site. The inhibitor enzyme bond is so strong that the inhibition cannot be reversed by the addition of excess substrate. That is in the case of irreversible inhibitor of inhibitory action of PMSF on serine proteases. Then deficiency of HGPRT and glucose 6-phosphate lead to. So HGPRT is hypoxanthine guanine phosphoribosyl transferase, and its deficiency is an inherited disease of purine metabolism, is usually associated with hyperuremia. The body's metabolism of glycogen is blocked by the absence of the enzyme glucose 6-phosphatase, which regulates the release of the glucose from glycogen stored in the liver. This results in an abnormal accumulation of glycogen in liver, causing liver to enlarge and produce symptoms of hypoglycemia, that is low blood sugar, and hyperuremia, that is gout. So here deficiency of HGPRT and glucose 6-phosphate lead to gout. Answer is option C. Then which is not an RNA virus? So HPV, double-stranded DNA virus, human papilloma virus. It is not an RNA virus. Secondary metabolites are produced in which of the following stages? That is stationary phase. Answer is option C. Then which one of the following amino acid has pKa value near the physiological pH? That is histidine. Histidine is having pK value near to 7. 6 point something it is. I don't remember exactly. Okay. Somatic mutation of immunoglobulin gene accounts for. So somatic mutation is found in immunoglobulins and it accounts for affinity maturation. Answer is option C. This question was repeated for I think for some other PhD entrance examination questions. You can refer to them. I have already made some videos on PhD entrance examinations for Delhi University, JNU and all. You can refer that. Hyderabad University also I have done. Then, many microorganisms can't use carbon dioxide as their sole source of carbon because further reduction of carbon dioxide is not possible. That is why they cannot use carbon dioxide as their sole source of carbon. Then there are three groups of photosynthetic bacteria, the cyanobacteria, green bacteria and purple bacteria. Then in humans, the inherited autosomal recessive diseases, Xenoderma pigmentosum, is the result of a defect in. So it is the defect in repair of UV damaged DNA. Answer is option D. Then which one of the following methods requires multiple set of primers to detect more than one organism targeting multiple loci from the genomic DNA? So it is multiplex PCR. In multiplex PCR, more than one target sequence can be amplified by using multiple primer pairs in a reaction mixture. Answer is option C. Then the advantage of Edmond reagent phenyl isothiocyanate over Sanger's reagent FDNB in peptide analysis is. So the process can be repeated on the remaining peptide. That is the advantage of Edmond degradation over Sanger's reagent. Answer is option C. Then the transition state of a catalyzed reaction is a highly populated intermediate of the reaction pathway which is lower in energy than the reaction substrate. Answer is option C. Then ETC has a potential to produce highly reactive free radicals that can damage the cells. Which one of the following is useful to prevent oxidative damage in cell? It is glutathione peroxidase. Answer is B. Then gangliosides derived from glucosyl ceramide contain one or more molecules of sialic acid. DUMP is converted to TMP by. So thymidylate synthase catalyzes the conversion of deoxyuridine monophosphate to thymidylate TMP in a reductive methylation. So DUMP is converted to TMP by methylation. Answer is option A. Then next one. Esterification of cholesterol in plasma is catalyzed by. So plasma cholesterol esterification is catalyzed by the action of lecithin cholesterol acyl transferase on lipids on the surface of high density lipoproteins. So the answer here is option A, that is lecithin cholesterol acyl transferase. What makes stem cells different from other cells? So stem cells divides asymmetrically to give rise to daughter cells which remain as stem cell and a second daughter cell that divides and differentiate into one or more cell types. That is how stem cells differ from other cell types. So coming to the next question. Which is the correct hierarchy of gene activity in early Drosophila development? So it is maternal genes, then comes the gap genes, then pair rule genes and segment polarity genes. The next one. The lineage of all adult somatic cell lineages has been documented in Caenorhabditis elegans. Then Nobel Prize in Physiology and Medicine for the discovery that mature cells can be reprogrammed to become pluripotent was awarded to. So it was awarded to Sir John B. Gurdon and Shinya Yamanaka. Then with reference to spontaneous mutation, one of the questions asked by genetist was whether spontaneous mutations are induced in response to external stimuli or whether variants are present at a low frequency in most populations. In order to answer this, fluctuation test was carried out by. So the fluctuation test, it shows that mutation occur randomly in a growing culture and are not induced by the selection agent. And this was demonstrated by Luria and Delberg in bacteria. So the answer here is option A, Salvador Luria and Max Delberg. Then which one of the following experimental design is most suited to answer whether a newly discovered transposable element in yeast transposes through an mRNA intermediate? So retrotransposon, they transpose from one position to another in the cellular genome by means of an RNA intermediate. So this was demonstrated in the studies in yeast TY1 elements by Gerald Fink and his colleagues. So what they did was they placed a particular TY1 element called as TYH3 under the control of a GAL promoter so that its transcription as well as this transposition can be induced by adding galactose into the medium. Then what they did, they marked TYH3 with an intron. So after inducing transcription of TYH3, additional copies were found at new locations in the yeast strain. So that is, after adding galactose into the medium, there was transposition of TYH3 to new locations in the yeast strain. And when they examined it structurally, it was discovered that the introns have been removed from the RNA. So if RNA transcript is the intermediate in moving the TYH3, then it's subjected to splicing and the introns can be removed. Hence these results fit with the prediction that RNA mediated transposition is through mRNA intermediate. So here the answer will be introduce an intron which can be spliced out in the transposable element and test the presence or absence of the intron at a newly transposed site. Answer is option B. Then next one. With reference to lac operon, which of the following mirror-diploids will show constitutive expression of beta-galactosidase? Now before coming to the question, I will tell you that there is something called as OC. OC is a mutation in the operator sequence and reduces the repressor from recognizing and binding to the operator sequence. And OC mutants LAC, ZY and A are expressed constitutively. That is, whether lactose is present or lactose is not present, LAC, ZY, A, that is the structural genes, they will be expressed. So coming to the first option, that is option A. I minus O plus Z plus Y minus. I plus O plus Z minus Y plus. So this is the first. I have drawn it like this just for you to understand. Okay, now I minus. So repressor is not being produced. But in the F prime form, that is I plus. So repressor is produced and it can bind to the operator of this half as well as to the F prime half. And it will not result in the transcription and translation of the structural gene. So beta-galactosidase will not be produced. If you see option C, also operator is constitutive. So the repressor cannot bind to this particular half of the operator, but it can bind to the first half. So it will be It will not synthesize Z plus in the case of first half, whereas 1 minute. Yeah. So it will not synthesize the Z plus in the first half because the repressor can bind there. Then whereas since the operator is constitutive, repressor cannot bind there, but it is Z minus. So beta galactosidase will not be synthesized. Now if you see option D, then also you have got operator constitutive as well as repressor. The other half is F minus half is having the repressor, whereas here the first half is not having the repressor production. So but this repressor, it can bind to the operator sequence O plus but not to OC mutation. But here also it is Z minus, so XGAL will not be produced. And here since the repressor binds to the operator, structural genes will not be synthesized. But if you see option B, there is constitutive, operator is constitutive, the repressor is not produced in this half, and Z plus is expressed. It is Z plus, so that means beta galactosidase will be expressed. The other half will be I plus, so where the repressor can bind to the operator and there will not be expression as Z expression of the structural genes, and moreover it is Z minus. So the answer here is option B. Then in what way are the homeotic genes of a flowering plant similar to those of Drosophila? So mutation in the homeotic genes cause transformation of one organ to another. That is option C is the answer. Then integrin signaling promotes cell migration by inducing changes in the cytoskeletal organization. Answer is option C. Photon capturing prosthetic group moiety of chlorophyll molecule is magnesium porphyrin. Answer is option B. Then opsonization process is involved with. So opsonization process is involved with macrophages. Answer is option D. Poly-L-leucine solution in dioxane is. So it is alpha helix. Answer is option D. Then next one. Which one of the following is best suited for hydrolyzing peptide bonds on the carboxyl side of aromatic residues? So it is chymotrypsin. Answer is option A. Then next one. Which one of the following molecule has the highest diffusion coefficient in plasma membrane? So it is glycophorin. Answer is option B. Cystic fibrosis transmembrane regulator protein is A. So it is a chloride transporter. Answer is option C. Which one of the following modification target a protein for degradation? That is ubiquitination. Answer B. Regulation of blood glucose level is. So it is due to opposing effects of insulin and glucagon. So insulin allows the cell to absorb glucose from the blood, while glucagon triggers a release of stored glucose from the liver. So regulation of blood glucose is by opposing effects of insulin and glucagon. Then which of the proteins or polysaccharides to which fibronectins are capable of binding is in a normal plasma membrane component? So it is integrin. Answer is option D. The next one. The observation that plasma membrane protein mix after cell fusion provide evidence for the fluid mosaic model. Answer is option C. Then treatment of hepatocytes in vitro culture with ricin leads to. Ricin is highly toxic type 2 ribosome inactivating protein. So it will naturally result in inhibition of translation. Answer is option B. In gel filtration chromatography, the large protein will elute first. It will not enter into the pores. So it will come through the in-between area, in-between region of the pores. It will not enter the pores. So it will be the first to elute. Two-dimensional gel electrophoresis is a technique for separating protein based on both charge and mass. It is based on isoelectric focusing as well as SDS page. So that is two-dimensional gel electrophoresis. So it is based both on charge and mass. Then exact mass and sequence of proteins and peptides can be measured by using MALDI-TOF. That is matrix assisted laser desorption, ionization, time of flight, mass spectrometry. Answer is option A. Then fluorescence microscopy is based on the ability of certain molecules to absorb light of a given wavelength and then emit light of a longer wavelength. Answer is option C. The next one. An IgG and an IgM samples against human red blood cell surface antigen were treated with beta-mercaptoethanol independently and tried for agglutinating human red blood cells with the view of checking the heme agglutination titer of the two samples. Which one of the following results is correct in this contest? Now IgG and IgM samples, they were treated with mercaptoethanol independently. IgG was treated independently, IgM was treated independently. And then they was tried for agglutination of human red blood cells with the view to check heme agglutination titer of the two samples. Now out of this, what is the correct option? So IgM, it is a pentameric form and has more disulfide bonds than IgG. Beta-mercaptoethanol disrupts the disulfide bonds found in the protein. The added beta-mercaptoethanol disrupts disulfide bond, thereby enabling IgM to be inactive and permit the agglutination only by IgG antibodies that resist beta-mercaptoethanol. This I got from a research article. So the answer will be the titer of IgM was found to be drastically decreased in comparison with that of IgG remaining unaltered. Answer is option A. Then next one. Lectin can bind to plasma membrane vesicles if it is. So first, what is inside out? Inside out is a vesicle produced from plasma membrane by mechanical disruption that has a cytoplasmic phase of the membrane on the outside. And the right side out is in which the outer leaflet of the vesicle faces the buffer. That is just like an intact plasma membrane. That is right side out. Now carbohydrates, they are important part of membranes and they are an important facet of the extracellular matrix. Sorry. Lectins are proteins which bind to carbohydrates and carbohydrates are usually exposed on the cell surface and lectins bind to the carbohydrates. There are no glycoproteins on the internal side, therefore inside out vesicles do not interact with the lectin, but right side out vesicles do interact. So a lectin can bind to plasma membrane vesicle if it is right side out. Then membrane asymmetry can be regulated by. So membrane asymmetry is established and regulated by three classes of enzyme, that is flippase, floppase and scramblase. So here only option B is correct, that is they have given only flippase there. So option B is correct. What is the activated reactant in the biosynthesis of phosphatidylinositol from inositol? So the synthesis of phosphatidylinositol catalyzed by phosphatidylinositol synthase and involves CDP diacylglycerol and L-myoinositol. So answer is option B, CDP diacylglycerol. Best method in tracking the synthesis and maturation of a protein in a living cells is. So it can be, best method can be making a fusion protein with GFP. So GFP will help you to track the synthesis and maturation of proteins in the living cell. Next question. Fluorescence recovery after photobleaching is a powerful technique for calculating the diffusion coefficient of membrane lipids and proteins. Answer is option A. Then next one. The concept of magic bullet proposed long back for targeted anti-cancer drug delivery is not a reality till today because of the lack of avidin biotin type high affinity of anti-cancer antibody. The avidin biotin type complex, it is the strongest non-covalent interaction between protein and a ligand, and the bond once formed, it is unaffected by extremes of pH, temperature and organic solvents and other denaturing agents. So the concept of magic bullet was not a reality till today because of the lack of avidin biotin type high affinity of anti-cancer antibody. One of the major reasons of the failure of somatic gene therapy protocol can be assigned to. So in somatic gene therapy, the therapeutic gene are introduced into the somatic cell or body of a patient. Any modification and effect will be limited to the individual patient only and will not be inherited by the patient's offspring or future generations. So one of the major reasons of the failure of somatic gene therapy protocol can be assigned to lack of technique in site-specific integration of a foreign gene into the chromosome of target cells. Then SRP selectively recognizes endoplasmic reticulum signal sequences on newly synthesized proteins. This is an outcome of. So SRP is signal recognition particle, and when a signal recognition particle recognizes endoplasmic reticulum signal sequence, it guides the ribosome and its growing polypeptide chain to the endoplasmic reticulum. So the answer here is selectively recognizes, and this is an outcome of, this is an outcome of formation of covalent intermediate. Answer is option D. Then, in the active state of transaldolase, which amino acid forms Schiff's base? So it is lysine. Answer is option C. Then an enzyme has an activation energy of 10,700 calories per mole. How many times faster will the reaction proceed at 37 degrees Celsius compared to 15 degrees Celsius? So you have to use the formula log K2 by K1 is equal to EA by 2.303 R, T2 minus T1 and T2 by T1, where K1 is the rate constant of the reaction at temperature T1 and K2 is at temperature T2. EA is the activation energy and R is the gas constant, and upon substitution you will get 4 as the answer. So it is 4 times. The rate of reaction at 37 degrees Celsius will be 4 times to the rate of reaction at 15 degrees Celsius. Then identify the small protein 167 amino acid that is produced in adipocytes and act as receptors in the hypothalamus to curtail appetite. So it is leptin. Leptin is a hormone predominantly made by adipose cells and enterocytes in the small intestine, and that helps to regulate energy balance by inhibiting hunger, which is in turn diminishes fat storage in adipocytes. So the answer here is option D, that is leptin. A TLC run of rat liver phospholipid is sprayed with ninhydrin and the color is allowed to develop. Which phospholipids can be detected in this way? It is PS, that is phosphatidylserine. Serine is an amino acid is detected by ninhydrin. Answer is option C. The next one. N-ethyl malimide is known to selectively inhibit bacterial lactose transporters. Which amino acid of the transporter is responsible for this inhibition? So N-ethyl maleimide is reactive towards thiols and commonly used to modify cysteine residues in proteins and peptides. So the answer is option A, cysteine. Highest turnover number of an enzymatic reaction so far known is exhibited by. So it is exhibited by carbonic anhydrase. Answer is option D. The chemical nature of covalent linkage in a disaccharide is known as. Nature of a covalent linkage in disaccharide is known as. So glycosidic linkage is a type of covalent bond that is formed between two carbohydrate molecules and it is of the form ROR type, that is ROR linkage, and it is an ether linkage. Answer is option B. Cancer cells have the unique property to exhibit uncontrolled division. This is primarily due to loss of contact inhibition. Answer is option D. The process which inhibits the uncontrolled growth of normal cell due to its contact with the surrounding cell is called as contact inhibition, and the cancer cell has lost the property of contact inhibition and it shows uncontrolled division.